where we need to go and what we need to do is to be able to take these large multi-omics, lipidomics type studies where we see alterations in classes of lipids, take that more mechanistically, looking at the specific species that are being altered, so not just the untargeted metabolomics and lipidomics, but then take that further into targeted pathways and looking not only at the dysregulation of lipids, but which lipids are changing, how are they changing, and by what mechanism.
Sphingolipid role in immunity. I don't know, sphingolipid storage.
Well, from what we've heard today, I think it's about being able to unpack more complexity with all of the trifunctional lipids and different tools that are being developed and more specific inhibitors for the different enzymes.
Yes, I think the tools to modify sphingolipids in specific biosynthesis is going to be interesting in the future.
Translational approaches to EV studies.
Developing new sphingolipid enzyme inhibitors.
Ceramide localization in the cell in relation to cancer.
We need to understand the mechanisms by which the lipids function in the cellular processes.
Understanding the evolution of the sphingolipid biosynthetic pathway. How did we get from single cell organisms with basic sphingolipids to where we are now?
It's going to be a new drug that's going to be, yes, that's going to put us again in the map. Sphingolipids will be in the map again. I think that's what it is.
All right, the impact of diet on the complexity of sphingolipids.
To find the mechanisms of disease and also use mediated by sphingolipids, and also use sphingolipids as biomarkers and end products and use it for diagnosis, prognosis, and for treatment.
I don't know, sphingolipidomics. I need to refine it.
They're going to be when we can actually translate lipid biology more into clinical.
It's definitely knowing the potential of sphingolipid and its impact in cancer biology, both in diagnosis, prognosis, and also to understand the resistance mechanism.
Protein-lipid interactions.
Hello, everyone. I believe the next great frontier in sphingolipid biology should be application to medicine, and in particular cancer.
Sex differences in sphingolipid metabolism.
How to visualize sphingolipids and by movement, for me, will be a really good question.
So as a systemic understanding of the whole network at the organism level.
Deoxysphingolipids.
Biomarker for multiple sclerosis.
Glycosphingolipids.
Use sphingolipid biology to understand evolutionary relationship.
It is to try to figure out what each species of sphingolipid is doing, particularly in terms of ceramide species, other glycosphingolipid species. I think the technology right now, we analyze many species by mass spectrometry, and we need to think about the next technology that can help is that.
Research probably are more going the direction of cell biology, trying to understand what sphingolipids do in the context of organelles, in the context of development, and in the context of cell-to-cell interaction, and how all of the above works together.
I don't know what the next frontier is, but I know that the people that are going to be involved are here at this meeting.
Identifying targets of ceramide. How about you? I drank too much. I have no idea.
So Basim, what's the best discovery at this conference?
This was a great meeting. I learned a lot. But the best discovery I have in this meeting is Bulgarian brandy, guys. Best!